Let's begin with a very real situation. You have a peptide sequence in front of you. The biology looks promising. The therapeutic idea makes sense. But before anything moves forward, one critical question comes up. How should this peptide actually be synthesized? Solid phase or liquid phase? This decision may look academic at first, but in real projects, it can decide whether you finish on time, stay within budget, or end up repeating weeks of work. In this presentation, we'll walk through both approaches, not just what they are, but when and why experienced teams choose one over the other. At its core, peptide synthesis is about precision. We take individual amino acids like alanine, serine, lysine, and chemically connect them in a very specific order. That order is everything. One missing amino acid, one incorrect linkage, and the peptide may lose activity entirely. This precision is why peptide synthesis sits at the heart of drug development, research tools, diagnostic assays. In industry, peptide synthesis isn't just chemistry. It's the bridge between biological ideas and real, testable molecules. Now let's talk about solid-phase peptide synthesis, or SPPS. In SPPS, the peptide chain is built while attached to a solid, insoluble resin. You can think of the resin as a stable anchor that holds the growing peptide in place. Each cycle follows a predictable rhythm. First, an amino acid is added. Then, excess reagents are washed away. Next, protecting groups are removed. And the cycle repeats. The key advantage here is that the peptide stays put while impurities leave. This simple idea is what makes SPPS so powerful. In real labs, SPPS wins because it saves time and reduces complexity. Because purification happens mainly at the end. Reaction cycles are faster. Manual handling is reduced. Automation becomes practical. This is why SPPS is widely used for therapeutic peptides, diagnostic peptides, peptide-based vaccines. From milligram discovery batches to gram-scale production, SPPS offers consistency, reproducibility, and speed. 3 things development teams value deeply. Liquid-phase peptide synthesis takes a very different approach. Here, everything happens in solution. There is no resin, no solid support. Each amino acid is coupled, then purified, before the next one is added. This means every intermediate can be isolated, analyzed, and fully understood. For chemists who want maximum control, LPPS can feel very elegant. But that control comes with trade-offs. LPPS shines when working with short peptides, especially dipeptides or tripeptides. Its strengths include high coupling efficiency, easy isolation of intermediates, lower consumable costs for small sequences. This makes LPPS particularly useful in structure, activity relationship studies, where understanding each intermediate matters. However, purification after every step makes LPPS time-consuming, solvent-intensive, difficult to automate. As peptide length increases, LPPS quickly becomes impractical. When comparing the 2 methods side by side, the pattern becomes clear. SPPS is built for longer peptides, high throughput, automation, and scale up. LPPS is better suited for very short peptides, detailed mechanistic studies, situations where intermediate control is critical. Neither method is universally better. The best method is the one that matches the actual project goal. Let's look at a real example. A clinical stage biotech company needed a 25A amino acid antimicrobial peptide. The requirements were strict: nearly 99% purity, reliable yield, tight timelines. Using solid-phase synthesis, the peptide was delivered in just 19 working days. The final batch achieved 98.7% purity by HPLC, 285 milligrams of reproducible yield. Because synthesis was efficient, formulation studies could begin early, saving valuable development time. This is a case where SPPS wasn't just preferred, it was essential. So, how do experienced teams decide? Choose SPPS when the peptide is longer than 10 amino acids. Speed and scalability matter. Automation is required. You're working on therapeutic or diagnostic products. Choose LPPS when the sequence is very short. Intermediate isolation is critical. Cost efficiency matters for small molecules. You're doing SAR or mechanistic chemistry. With proper purification and characterization, both methods can meet pharmaceutical standards. In the end, choosing a peptide synthesis method is about balance. You balance length and complexity, timeline pressures, scale and purity needs, regulatory expectations. The smartest teams don't ask which method is better. They ask which method serves this peptide best. That mindset is what turns peptide chemistry into successful drug development. If you're evaluating peptide synthesis strategies and want guidance grounded in real-world experience, Resolve Mass Laboratories helps teams make the right choice right from the start. Because in peptide development, the right synthesis decision doesn't just save time. It accelerates success.